All right, so welcome back. This is going to be our screencast over Chapter 8. And in Chapter 8, we are going to be looking at the concept called metabolism. When you think about metabolism, what you want to do is you want to think about all of the different types of chemical reactions that are occurring within your body, and you want to think about how those chemical reactions actually occur. And that's what Chapter 8 is going to discuss. So when you think about, again, an organism's metabolism, what you want to think about is you want to think about something called transformation. You want to think about the things that are being produced, and you want to think about the things that are being torn apart. And in order for those things to occur, you need to have a source of energy, because without that energy, you will not be able to perform the work that is necessary to either create or dismantle those things that are required by a living organism. So when you think about metabolism, you want to think about something called the totality of the organism's chemical reactions. And what that totality means is every single chemical reaction that would be occurring inside of that cell to make sure that that cell remains healthy and is able to support that living organism. So when you think about metabolism, you think about various different pathways that will actually be used to produce those chemical reactions. And when you think about a pathway, typically you begin with a specific molecule, and then of course you will end with some sort of product. So each step is going to be catalyzed typically by a specific type of enzyme. And remember back to our discussion of macromolecules, an enzyme is considered a protein. So there's lots of different enzymes that you would find within our body. Down here towards the bottom, what you're noticing is that this is a good example of a metabolic pathway. And our starting molecule is going to be molecule A. And the product that we're going to finish up with is going to be product D. Now you'll notice that there are two intermediates between A and D. So we have intermediate B and we have intermediate C. And the only way you can get to these two intermediates is by using these specific enzymes to basically catalyze or create the reaction that is necessary to produce product B. And the same thing goes for product B to product C. So again, you need enzyme 2 to be able to produce product C. Then of course when you get ready to produce our final product, which is what we're shooting for, product D, you're going to need the work of enzyme 3 to be able to produce that product. So there are two types of metabolic pathways that we tend to focus on in biology, and the first one is called a catabolic pathway. And when you talk about catabolic pathways, you're talking about those pathways that will release energy, and they do this by breaking down complex molecules into simpler compounds. A good example of this would be cell respiration, and that's something that we had talked about back in Honors Bio, and we will talk about in AP Bio. And cell respiration is simply taking glucose and putting that glucose in the presence of oxygen, so the oxygen that we take in, and through a series of very complex chemical reactions, we are going to break down that glucose and actually produce ATP. And again, there's a specific pathway that is necessary to end up with that end product of ATP. And when you think about the second type of metabolic pathway that we would focus on in biology, it's called an anabolic pathway. Now in this particular pathway, we are going to be consuming energy. Instead of releasing, like we had seen in catabolic pathways, we are going to consume energy to build complex molecules from simpler ones. And a really good example of this one would be the synthesis or the making of protein from amino acids. Remember that a protein is considered a polymer, and in order to produce that polymer, we need to have those very tiny bits called monomers. And the monomers to build a protein are going to be the amino acids. And to basically put those amino acids together, we would need the anabolic pathway to be able to do that. Now, as we had said earlier, in order for these pathways to actually proceed, there needs to be energy put into the system. And when you think about energy, energy is simply the capacity to cause change of some sort. Energy is going to exist in various forms, and some of which can perform work for us. The four primary forms of energy that we would focus on in AP Bio would be something called kinetic energy. And if you think back to middle school when you maybe took physical science, your teacher might have mentioned something called kinetic energy, and this is considered the energy of motion. So anytime that you are moving around, for example, as I move my pen around, that would be a good example of kinetic energy. The second would be heat or thermal energy, and this is going to be any kinetic energy that is going to be associated with the random movement of atoms or molecules. The third is going to be potential energy, and this is going to be the energy that matter possesses because of its location or structure. A good example oftentimes that teachers will use to sort of illustrate this would be looking at, for example, a slide. And if you put a ball at the very top of that slide, and you will notice that at this particular height we have a large amount of potential energy. And then if we push that ball and it makes its way down the slide, when it ends up at the very bottom, we're going to have a ball that's going to have a very small amount of potential energy. So it's basically energy of position is what that's going to be. Then we have something called chemical energy, and this is the type of energy oftentimes that we are either using or producing inside of our cells. And this is going to be potential energy that's going to be available for release in a chemical reaction. Now this energy can be converted from one form to another. A good example of this, again, is going to be light energy that is used during photosynthesis. We can't directly use light energy, so what we have to do is we need a process that's going to convert that light energy into usable chemical energy, and that's going to be stored in the bonds of the glucose that's produced during photosynthesis. So if you think about the information that I presented in the previous slide, we had talked about the idea of taking light energy and actually changing it into a form that cells could actually use, which would be a form of chemical energy. And so that would be a good example of energy transformation. Well, there's actually a word that we would give to the study of this transformation, and that's going to be called thermodynamics. Now, when you talk about living systems, or if you talk about any system, living or non-living, typically we place them into two categories. The first is something called a closed system, and a good way to illustrate this would be a thermos. For example, the hydro flask that I have here, this would be considered a closed system. The idea is that whatever I put into this thermos, the energy most of the time would be in the form of heat, would be contained within this thermos. There's no way for it to get out into the environment. The second system that we would look at would be something called an open system. And this is going to be a system where energy and matter can actually be transferred between the system and its surroundings. And any living organism on this planet is going to be part of an open system. So we have two different laws of thermodynamics. So the first law of thermodynamics is going to be basically the energy of the universe is considered constant. So what that basically means is that energy can be transferred. In other words, you can move it to a different place. You can transform it. Maybe as you move it, you change it into a different form of energy. But there is no way for you to create energy or to destroy that energy. So it's simply going to change form. Sometimes teachers will actually present this law of thermodynamics as the principle of conservation of energy, which basically means it's conserved. It stays around. It just might be moved into a different form. And down here towards the bottom, this is a good example of the first law of thermodynamics. As we had said, when you talk about photosynthesis, you talk about plants that will take the light energy from the sun and basically change it into a form that we could use, which would be chemical energy. And that chemical energy is going to be stored within the food that we eat. Then, of course, we're going to take that food and we're going to use the energy produced from that food to provide us with kinetic energy, or that energy of motion. And then as we move around, we're going to produce heat, body heat. And that's going to be, again, a different form of energy, and that's going to be called thermal energy. So the first law of thermodynamics, again, is that we cannot create or destroy energy. The only thing we can do is move it around, transfer it, and possibly transform that energy. Now, the second law of thermodynamics basically states that every energy transfer or transformation is going to increase the entropy or the disorder of the universe or the environment. And the easiest way to sort of understand the second law of thermodynamics is to look at this illustration of diffusion. So this is what we had looked at back in chapter 7. So on the left-hand side of the diagram, you're going to notice that we have a relatively organized set of molecules. And in this case, it's going to be molecules of dye. and we have a membrane that's going to be down right through the middle of our container, but again, it's going to be a porous membrane. So there are areas within this membrane that these molecules can escape from. So over a period of time, these molecules are going to make their way through these pores in the membrane and eventually reach a state of equilibrium. So what has happened is that before we had a relatively organized area of molecules on the left-hand side, but what they did is they actually distributed themselves throughout the container as they moved through these pores. And that's going to basically demonstrate the idea of disorder. In other words, they don't like to stay in one place. Because of the random motion of the molecules, they made their way out of the left-hand side and into the right-hand side of the container until they reached a state of equilibrium. Now, when you talk about energy, oftentimes when you're a scientist, you want to know whether or not the reactions that you will be working with are going to occur spontaneously and which ones are going to require the input of energy in order for them to actually proceed. Obviously, the ones that occur spontaneously are going to be the ones that we're going to be a little bit fearful of because those are going to be the ones that could be pretty dangerous. And then, of course, if we're trying to get a particular reaction to proceed to produce a product, we need to know what type of energy do we need to put into the system to get that chemical reaction to go. So we need to determine what we call energy changes that occur in these chemical reactions. So when you talk about a living system, you oftentimes talk about something called free energy. Sometimes instead of calling it free energy, sometimes it's easier to think about it as available energy. All right, so that available energy that could actually be used by the cell to do work. And that's going to be that energy that's available when the temperature and the pressure are relatively uniform in the cell. So the change in free energy, which is often referred to as delta G, during a process is often related to a change in what we call enthalpy, which is often identified by delta H and described as the total energy that you would find in the system. A change in entropy, which we had talked about in the previous slide, which is going to be identified by delta S, and so again that's sort of that chaotic or disorder idea within the universe, and of course temperature, which for our case we're going to identify that in the units of Kelvin. Now this is an equation that you will often see both in bio and in other types of sciences. This basically states that delta G is going to be equal to the total energy minus temperature combined with that entropy that we had just talked about. So one way to think about this is if you think about the total energy in the system, we need to understand that in order for any type of reaction to be spontaneous, we need to make sure that that delta G, or that free energy or available energy to the cell, is in a negative state. And when you think about the total energy in the system, if you were to bring down the total energy of that system, it would make sense from this equation that you would also bring down free energy. And if you look at these two, let's say for example we decided to, if you notice we have a negative here, if we decided to increase the temperature or maybe increase the disorder in the system, that also would actually bring down the delta G, or the free energy. In other words, that's going to make it more negative. So that's going to sort of encourage the idea that the chemical reaction is going to occur spontaneously. And spontaneous processes can be harnessed, or basically the energy that they produce can be used to perform the work that we need to make sure that we get things done. So when you think about the free energy, again, oftentimes what we'll do is we'll think about the idea of stability, and because stability is important when it comes down to living things, and of course equilibrium. So free energy is going to be the measure of the system's instability. In other words, its tendency to change to a more stable state during a spontaneous change. In other words, if we have a spontaneous reaction occur, that free energy, again, that delta G that we had just talked about, which would be negative because it's a spontaneous change, is actually going to decrease, and the stability of the system is going to increase. So let's say, for example, it was -5 for delta G, then it's going to proceed, for example, to maybe -2 or maybe even 0. It could be even a positive number. And that's going to sort of encourage the idea of stability and equilibrium within the system. So a process is spontaneous and actually can perform work only when it's moving towards equilibrium. So again, more free energy, higher G, less stable, greater work capacity. So that's at the very beginning. In a spontaneous change, the free energy of the system is going to decrease. In other words, the negative g, the system is going to become more stable. Remember, it's going to kind of shoot towards that stability factor. The released free energy can be harnessed to do the work, and at the very end, less free energy, lower g, more stability, less work capacity. So in other words, we sort of use up that energy as we perform the work. So what we have down here is we actually have three different situations where we sort of demonstrate that. Up here, if you think about the amount of free energy, the amount of potential that we have up here at the very top, as you dive off of the platform, the kinetic energy, of course, is going to take over. And we have lots of kinetic energy at the very beginning, makes its way down to the water, and then we're kind of heading towards more of a stable situation. Once that diver hits the water, stability is going to ensure. And so basically that delta G is going to become more positive. So when you look at this example of diffusion in the middle, what you're going to notice is all of these molecules are in one area, but what they want to do is they actually want to spread out. And so what's going to happen is you're going to notice they're going to, again, be kind of moving towards that idea of stability or equilibrium within that system. And that's going to cause that delta G to become more positive. And the same thing happens here with this molecule during a chemical reaction. If you have a lot of energy stored within these bonds, if it's a spontaneous reaction, again, a very high delta G, in other words, more negative number. But then what's going to happen is once that reaction occurs and we get the products at the very end, again, we're heading towards that state of stability. So that delta G is going to be lower or more positive. Now, when you think about free energy, you want to also think about it in relation to metabolism. So the concept of free energy can be applied to the chemistry of life's processes. And remember, metabolism is the totality of all the different chemical reactions that occur within a cell. So we have two different types of reactions that we would identify here. Something called an exergonic reaction, and that's going to proceed with a net release of free energy. And again, it's oftentimes considered spontaneous. Then we have something called an endergonic reaction. That's going to absorb free energy from its surroundings. And because it absorbs energy, it is non-spontaneous. In other words, does not occur unpredictably. Now over here on the right, this is an example of an exergonic reaction. And so we have the free energy on the left. We have the progress of the reaction down here towards the bottom. So in an exergonic reaction, we have lots of energy stored up here. So if you look at the reactants, lots of free energy. It's very high. But because it's a spontaneous reaction, that energy is released pretty quickly. And what you have at the very end in these products is going to be relatively small. So again, the amount of energy released kind of indicates that delta G is going to be negative. But if you look at an endergonic reaction, remember that the amount of energy that you would find in the reactants is going to be rather small. You need to absorb energy from your surroundings. Again, it's endergonic. And what you're going to find is that at the very end, again, through the progress of the reaction, the amount of energy in the products is going to be relatively high. And this is going to indicate a more positive delta G. Now, equilibrium is really important when it comes down to metabolism. So reactions in a closed system do eventually reach equilibrium. Now remember, we are actually an open system, but what you want to do is you want to make a comparison to a closed system. So the reactions in a closed system eventually do reach equilibrium, but the problem with that is then what happens is they are not able to do any work. Cells are not considered to be in equilibrium. So like I had said, we are an open system. They are an open system experiencing a constant flow of materials both in and out of the cell. So a defining feature of life is that metabolism will never be at equilibrium. So your body is always working really hard to do its best to keep it in a relative state of equilibrium, but it's never going to be completely equal. So a catabolic pathway in a cell is going to release energy in a series of reactions to sort of encourage the idea that our reactions can continue or keep going. Now, one of the ways that oftentimes they will sort of illustrate this is that they will use this hydroelectric system on the right-hand side. And so if you notice, in an isolated hydroelectric system, in other words, it's a closed system, over a period of time, a hydroelectric system is going to provide power. Over a period of time, if you go from this area of the system to this area, the water is going to flow downwards. And so we're going to get this turbine that's going to actually turn, and that's going to provide us with power. But because it's a closed system, eventually there's going to be an equal amount, relatively, of water on each side, and that turbine is going to stop. So if you notice, in the beginning, delta G was negative. In other words, it's going to have a lot of free energy. But then if you notice over here on the right, delta G is now 0. So it's kind of reached equilibrium and the turbine is no longer turning. So that's not a good system to have, especially when it comes down to living organisms. But if you take it and you put it into an open type of system arrangement where you have water that is moving into the system and water is flowing out, then you sort of encourage that I don't want to use their word instability, but that openness of the system. And if you notice, that turbine will continue to move, and that's going to provide us with the electricity that we need. And that's going to encourage the negative aspect of free energy. And down here towards the bottom, this is just another example and shows a multi-step open hydroelectric system. So in this case, we have 3 turbines that are being turned, but again, water is coming in, water is leaving, and so you have the constant motion of the water, which is maintaining the free energy as being negative for each of these 3 different situations. So when you talk about energy, you cannot get away from talking about ATP because ATP actually powers cellular work. And a cell does 3 main kinds of work. It's going to do a chemical type of work, it's going to do a transport type of work, and it's going to do a mechanical type of work. Chemical, of course, represents the different chemical reactions that occur within our body. Transport, for example, transport of substances across the cell membrane is a very important type of work. And then we have mechanical, or the ability to move things around. And again, that's a very important type of work when it comes down to living things. To do work, cells are going to manage their energy resources by something called energy coupling. And this is going to be the use of an exergonic, in other words, spontaneous or energy producing process to help drive an endergonic or energy absorbing type of process. So most energy coupling in cells is going to be mediated or determined by ATP. And remember ATP is adenosine triphosphate. And this is going to be the cell's energy shuttle, in other words, to move energy around the cell. And up here is a good example of the ATP molecule. And this is something you had looked at again back in Honors Bio. So when you think about the ATP molecule, you think about basically 3 parts. You think about the ribose, again that 5-carbon sugar. You think about the phosphate groups, typically 3, again adenosine triphosphate. And then you think about this base up here, which is the adenine group. And those are the 3 parts that you would need to be aware of because they're going to be important when it comes down to releasing the energy that is going to be found in ATP. So the bonds between the phosphate groups of ATP's tail can be broken by hydrolysis, in other words, the addition of water. So down here towards the bottom, this would be the ATP molecule, and if we add water to the system, remember hydrolysis will add water, that's going to cause a break in the bonds between this first and second phosphate group. So the energy is going to be released from ATP when that terminal phosphate bond is broken. So this release of energy comes from the chemical change to a state of lower, again, free energy, not from the phosphate bonds themselves. So the energy is stored within the bonds, but again, because we release the energy, and again we're heading towards that state of stability, that delta G or free energy is actually going to go down. In other words, we're not going to have a high negative number, it's going to come down to what we would consider, I guess, a lower negative number. So how ATP actually performs its work, again, remember we talked about that work can be in the form of mechanical, transport, or chemical. This is going to be powered by the hydrolysis of ATP, like we had said in the previous slide. In the cell, the energy from the exergonic reaction, again, energy releasing of ATP hydrolysis, is going to be used or coupled with the endergonic reaction. So overall, the coupled reactions for whatever set of reactions we're looking at will end up being exergonic. And that's important because we need it to be able to produce that energy necessary for the change to occur. So over here on the right, what you're going to notice is that this is going to be the formation of glutamine, which is an amino acid. And to form glutamine, through an endergonic reaction, you have glutamic acid combining with ammonia. But the problem is it's really hard to do that because if you notice, the amount of free energy is in the positive state. And so in other words, it's endergonic, it's energy absorbing. And so what we have to do is we have to couple it with ATP. So we have ATP, which is going to phosphorylate, and we're going to talk about that in a second, glutamic acid, and that's going to make the amino acid a little bit less stable. So when you add ATP to glutamic acid, you actually add a phosphate group, again that phosphate came from the ATP, to the glutamic acid. So the ammonia is going to displace the phosphate group, and actually form the glutamine. So if you notice, that energy is going to be encouraging basically the glutamic acid and the ammonia that you see right here to actually come together. And so that's going to encourage that reaction to happen. So because of that additional energy, you're going to notice that in the very end, our numbers actually became negative. So remember, this was an endergonic reaction, and it's endergonic because the delta G, or free energy, is going to be positive. We want it to be negative, but we can't get there unless we actually contribute a little bit of energy. So that's where the ATP comes in. And so when we added that ATP, which is going to be about negative 7.3 kilocalories, lots of energy stored up there. If you do a simple subtraction, what you're going to end up with is, at the very end, a net of negative 3.9 kilocalories per mole. So that's the overall, again, free energy change, which is going to encourage the formation of that glutamine. So ATP is going to drive, again, the endergonic reactions, again, through phosphorylation. And again, phosphorylation was simply the transfer of a phosphate group to some other molecule. So remember, in the previous slide, we transferred that phosphate group to the glutamic acid to be able to help us produce the glutamine at the very end. So the recipient molecule is often called a phosphorylated type of molecule. So down here towards the bottom, we have two different types of work. We have transport, we have mechanical work. And in transport work, like you see here, and I think this is representing the sodium-potassium pump, what we're trying to do is we're trying to get molecules across the cell membrane. And if you notice, we have a P here, so we have a little bit of ATP that's being used, and that phosphate is actually being used to encourage the movement of these molecules. So again, phosphorylation has occurred, the transfer of the phosphate. And then if you notice down here towards the bottom, we have mechanical work. And here's going to be a good example of a motor protein. And if you notice, in order to allow that motor protein to walk along those microfilaments or microtubules, you need to provide a little bit of ATP. So again, the transfer of that phosphate group to make sure that energy is released to be able to cause that motor protein to do what it needs to do. In other words, to walk along that cytoskeletal track. So the regeneration of ATP is also very important because we do consider ATP a renewable resource. And it's going to be regenerated by the addition of the phosphate group that it had lost during phosphorylation. So the energy to phosphorylate ADP is going to come from various catabolic reactions in the cell. So remember, we have two different types of reactions. We have catabolic and we have anabolic. So when you think about catabolic reactions, you think about basically taking something and breaking it down. And so in a catabolic reaction, for example, like the breakdown of sugar, for example, glucose, because we break down that sugar, there's going to be energy stored within those bonds, and that's going to sort of drive the formation of the ATP. So if you look down here, the energy from catabolism is going to be exergonic, again, energy releasing, and that's going to come maybe from sugar, and that's going to encourage the attachment of this phosphate to this molecule of ADP, and it kind of reinvigorates it. In other words, it restores it to its original form, and so that's going to be the ATP that you see here. And then that ATP, of course, can be used to do work for other types of reactions, and that, of course, is going to be an endergonic or energy-consuming process. So when you talk about energy and you talk about metabolism, you need to make sure that you have a good understanding of enzymes, because these enzymes are going to basically be the chemical agent that is going to be used to speed up a reaction. Now, an enzyme is considered a catalyst, and there's actually two different types of catalysts out there. You have those that are organic, like enzymes. Then there's those out there that are considered inorganic, and those would be the ones that you might encounter maybe in a chemistry class. But in our case, we're going to be focusing on enzymes. So again, enzyme is considered a catalytic protein. Remember, it does fall into the category of proteins. A good example of the use of an enzyme would be the hydrolysis of sucrose. And remember, sucrose is simply a disaccharide, it's table sugar. And the enzyme that would be used is called sucrase. And you know it's an enzyme because it has the ASE at the very end. And so what's going to happen is this sucrase, again, by the addition of water, is going to be used to break this sucrose molecule into glucose and to fructose. And so that's going to be something that's going to be used to encourage the breakdown of that particular type of carbohydrate. So when you talk about enzymes, you need to understand that every chemical reaction out there between different molecules is going to involve some sort of bond breaking or could be bond forming. So the initial energy necessary to start a chemical reaction is considered the activation energy, sometimes called the free energy of activation. And this is the way that we would actually identify activation energy. So activation energy is often supplied in the form of heat, and it's going to be from the surroundings typically. And so down here towards the bottom, you're going to see this diagram that sort of illustrates the idea of activation energy. We have the progress of the reaction, and this would be considered the free energy in the reaction. And so again, free energy is identified by delta G. And so if you notice in this case, we have A, B, C, and D, and so we're trying to get to our final product of D, or maybe our final product is going to be A and C and B and D. And so if you notice, the reactants are going to be A and B together and C and D together. We put them together, but we need to encourage them to actually react and to sort of change positions and become A, C, and B, D. And in order to do that, we need to throw some energy into the system. So this right here would be considered the activation energy necessary to get this reaction to proceed. And so this right here is the amount that we need. Once that is provided, we sort of get over the hill and we end up with the products that you see down here at the very bottom. So this would be the amount of free energy that you would find in the system after the reaction has occurred. And if again you notice, it's at 0, which as we said before is an indicator of stability within that system. So when you think about enzymes, an enzyme is important because what it does is it actually helps to lower the activation energy barrier. Enzymes do not affect the change in free energy. What they do is they actually hasten or speed up the reactions that would occur naturally. So again, looking at our diagram down here, this is what would have occurred normally without the enzyme. Now if you notice there, without the enzyme, we have a lot of activation energy that is necessary to kind of get over the hill. But if we basically couple or provide an enzyme for this reaction, it's going to help to hasten or speed it up. It's going to take that activation energy and actually make it much less. So it's going to lower the amount of activation energy necessary to get the reaction to actually proceed. And so if you notice, this is going to be the delta G is going to be unaffected by the enzyme. So we're still getting to the same point, only we're able to get to that same point a little quicker with the enzyme. So when you talk about enzymes, of course, you need to have a good idea as to how they work. And so the reactant that the enzyme is going to act on is going to be called the enzyme substrate. And we're going to look at that in a little bit more detail in a second in a diagram. The enzyme is going to bind to this substrate, and again, substrate is going to be like the reactants, and it's going to form something called an enzyme substrate complex. Now the active site is going to be the region on the enzyme where the substrate actually binds. And we call that an induced fit. And so this is going to bring basically the chemical groups of the active site into the positions that are going to enhance or encourage their ability to catalyze the reaction. So down here towards the bottom, this is sort of a computer-generated example of an enzyme. So this blue area that you see right here, that's the enzyme. This is going to be the active site right through here. Again, the substrate is going to be the chemical reactants of our chemical reaction, and they're going to fit perfectly right within this active site. And so this would be considered, once it's there, the enzyme substrate complex. So when you think about an enzymatic reaction, in other words, a reaction that's being encouraged by an enzyme, remember the substrate is going to bind to the active site of the enzyme. Now the active site is going to lower an activation energy barrier. Again, remember we're trying to lower that activation energy by orienting the substrates correctly, in other words, making sure those reactants are in good proximity of each other. Straining those substrate bonds, in other words, remember when you talk about chemical reactions, you're talking about either forming or breaking bonds. Provide a favorable microenvironment. In other words, make sure again they're in close proximity and, as we had said, positioned correctly and covalently bonding to the substrate. In other words, making sure bonding happens correctly. So over here you're going to notice that the enzymes are going to have what we call an induced fit within that enzyme. We have substrates that are going to be held in the active site by weak interactions, and it could be something as simple as either hydrogen or ionic bonds. Then you're going to notice that the active site is going to again lower the activation energy. It's going to speed up the chemical reaction. The substrates are going to be converted to products. Remember, we're trying to get to an end product here. and then these products are going to be released into the environment. And so now the active site is available for two new substrate molecules. And what's kind of interesting about enzymes is they are never used up, and you can use these enzymes over and over and over again. So there are going to be some environmental factors out there that will affect an enzyme's activity. One of those will be temperature, and the other one is going to be pH. Chemicals that specifically influence an enzyme are also some things that we can discuss when it comes down to enzyme activity. So each enzyme has what we call an optimal or best temperature at which it can function, and there's also an optimal or best pH in which it can function. So down here towards the bottom, these are two graphs that sort of illustrate that. So there's two enzymes being represented here. This is the optimal temperature for a typical human enzyme, and if you notice, it tends to hover right around 37 degrees Celsius. So this is an optimal temperature for an enzyme of a thermophilic heat-tolerant bacteria. And again, this is going to be very organism specific. So in this case, for this bacteria, the optimal temperature for the enzymes in this particular organism are going to be around, I would guess, around 78 degrees Celsius. And again, it is a heat-tolerant bacteria, so it's going to be able to work at higher temperatures. So again, when you think about pH, optimal pH for pepsin, which is going to be an enzyme that you would find in the stomach, is going to be around a pH of around maybe 2, maybe a little bit less than 2. But if you notice here, optimal pH for trypsin, which is an intestinal enzyme, is going to be a little bit higher, and it typically hovers around 8. So when you think about enzymes, we need to make sure that we understand that we need to regulate the activity of these enzymes to help control metabolism. Chemical chaos, is the way they describe it, it would result if a cell's metabolic pathways were not tightly regulated. A cell does this by switching on or off the genes that actually encode specific enzymes, or by regulating the activity of the enzymes. So a word I want you to be aware of is something called allosteric. Allosteric regulation is either going to inhibit or it's going to stimulate or encourage an enzyme's activity. Allosteric regulation occurs when a regulatory molecule binds to a protein at one site and affects the protein's function at another site. And we're going to see a diagram in a second that's going to illustrate this. Most allosterically regulated enzymes are going to be made from polypeptide subunits, so in other words, polymers or proteins. Each enzyme has active and inactive forms. The binding of an activator is going to work to stabilize the active form of the enzyme, and the binding of an inhibitor is going to stabilize the inactive form of the enzyme. So in this diagram, what it's trying to show you is the difference between an allosteric activator and an allosteric inhibitor. So if you notice, this is considered the active form of the enzyme, and you're going to notice there's two sites. We have the allosteric enzyme, which is this right here. This, the whole thing is the allosteric enzyme, but we have 4 different subunits for that enzyme. And this is going to be considered the regulatory site. This is the site that actually will be there to actually hold on to the activator. In other words, make sure that we stabilize or encourage that enzyme to remain stable so it can do what it needs to do. And this activator is going to fit right about here, and you notice they put one here to make sure that it does remain stabilized. And again, down here towards the bottom, you're going to notice that we have an inactive form of this particular enzyme as well. This is a non-functional active site. In other words, it can't really do what it's supposed to. And the reason it can't do what it's supposed to is because this is considered the inhibitor. So in this case, this inhibitor is going to basically fit perfectly right through here, and that's going to stabilize the inactive form. So what that means is that we could have enzymes that are going to oscillate or basically move between the active form and the inactive form. And the reason for that is sometimes enzymes are going to be used to again, encourage these chemical reactions to produce a product at the very end, maybe something that we need a lot of, but maybe we get too much of it. And so what we need to do is we need to sort of oscillate back to the inactive form and maybe slow down the production of whatever that product might be. And these activators and inhibitors are going to work to encourage that to happen. Now, sometimes we have something called cooperativity, and this is going to be a form of allosteric regulation that might be used to actually amplify enzyme activity. In other words, encourage it for whatever reason. And this is going to be the binding of a substrate to one active site that's going to stabilize the favorable conformation changes of all the other subunits. And so this, again, cooperativity is another form of allosteric activation. So in this case right here, this substrate is going to bind to this particular area of the enzyme. So normally this would be considered the active form, but we're going to basically encourage or produce a favorable situation to stabilize the active form of the enzyme so we can continue to produce the product that we need. So the last thing that we need to do is we need to talk about something called feedback inhibition. And when you think about feedback, you're talking about something that's going to basically encourage or to give us information on whether or not we need to proceed with a reaction. Feedback inhibition is going to prevent a cell from wasting chemical resources by synthesizing more product than is needed. It's kind of similar to what we had talked about in the previous slide. So if you notice over here on the right, this is going to be a chemical reaction that's going to basically be encouraged by an enzyme. You notice we have enzyme 1. And in the beginning we have threonine, which is going to be a type of amino acid. And this threonine is going to fit perfectly within this active site. And so if it proceeds normally, then it's going to go through a series of steps. This is the metabolic pathway from A to B to C to D with the use of these different types of enzyme. And we're going to end up with isoleucine at the very end. So again, this is another type of amino acid. But sometimes we don't need as much of that end product, and so what we want to do is we want to introduce basically another molecule. And so this is going to be the allosteric site right here of this enzyme. And so if you notice, if we have a lot of isoleucine, something that could happen would be that the isoleucine will actually bind to the allosteric site. And again, when you think about it, when it binds to this site, it's going to sort of prevent or switch off the pathway. In other words, we're not going to get the production of that end product anymore. Now, if we use up the isoleucine, then what's going to happen is that this feedback inhibition is not going to be there anymore, and then we're going to get the encouragement again of this threonine to bind to this active site and start the process over again. In other words, the production of the end product isoleucine at the very end. Alright guys, so that's going to wrap up our discussion for Chapter 8. Again, make sure that you have completed your study guide for this chapter.